Greetings, students. The topic which we are going to cover is synthesis, storage, and destruction of histamine. So, histamine is a type of autocoid. Before we start histamine, let us see what is autocoids. Autocoid is made up of two words, autos meaning self and eikos meaning healing substance or remedies. So autocoids are substances which are self-healing substances. There are a wide variety of autocoids which are present in the body and which generally act locally on a particular site where they are synthesized and where they are released. The autocoids are also known as local hormones. Now, the reason they are called as local hormones is, first of all, we know that hormone, it is synthesized from a particular cell, from a specified cell. In the same way, our autocoid is also synthesized from a specific cell, and hormones, they are transported to some other parts and then they exert their activity. That is, they are synthesized in a particular part and they are having their activity in some other part of our body. But autocoids are synthesized and have activity on that particular site wherever they are released. That's why the term local is attached with hormones to specify autocoids. Now, these autocoids, they are broadly classified into three main types, that is amine autocoid, lipid-derived autocoid, and peptide autocoids. Amine autocoids include histamine and 5-hydroxytryptamine, which is also known as serotonin. Lipid-derived autocoids include prostaglandins, leukotrienes, and platelet-activating factors. Peptide autocoids include plasmakinins such as bradykinin, kallidine, and angiotensin. So, the histamine which we are going to study in detail is a type of amine autocoid. So, let us start with histamine. Now, again, if we break down the word histamine, it is made up of two parts, that is histose and amine. Histose is tissue. That's why histamine means tissue amine. So, they are mainly present in any specific animal tissues inside our body. In detail about histamine was studied by Dale in the year 20th century. So he gave the detail about histamine, that these histamine are actually released inside the body upon manifestation of any allergic reactions. These histamine, they are also released inside the body upon any hypersensitive phenomena or if there is any tissue injury inside our body. These histamine are mainly stored inside mast cells. Okay, so the tissues which are rich in histamine include skin, gastric, intestinal mucosa, lungs, liver, and placenta. Apart from that, they are also present in certain non-mast cells, such as in brain, epidermis, gastric mucosa, growing regions, etc. Histamine are also present in certain fluids inside our body, such as blood, in body secretions, in venoms, or in pathological fluids. Now, let us see how this histamine is synthesized, stored, and released inside our body. So, chemically, histamine is beta-imidazoline ethyl amine. So, if we see the structure, this is the structure of histamine as we can see. Okay, this is our amine group. Now we know that primary carbon attached to amine is known as alpha. The carbon attached to alpha carbon is known as beta. So on beta we have this imidazoline ring. That's why it is known as beta imidazoline. This is ethyl group and amine. So chemically this is beta imidazoline ethyl amine. Now, it is synthesized from a amino acid histidine and it is degraded by two pathways, that is oxidation and methylation. Now, we have already seen in the introductory slide that it is stored inside the mast cell. So this histamine, it is actually positively charged and therefore it requires any negative counterpart for its storage. So inside mast cell, this is stored along with negatively charged acidic protein or heparin. Now, this is the simplified version of synthesis and degradation pathway, as we have already seen that it is synthesized from amino acid histidine. Now, it is synthesized in presence of decarboxylase enzyme. And we have already seen that it is degraded by two pathways, methylation and oxidation. So methylation takes place in presence of N-methyl transferase and it results in N-methylhistamine. This N-methylhistamine, again in presence of monoamine oxidase, converts to methylimidazole acetic acid. So this is one pathway. Second pathway is oxidation, which takes place in presence of diamine oxidase. Diamine oxidase converts it into imidazole acetic acid. Now we will see the same pathway along with chemical structures. So this is the structure of histidine, which also has imidazolyl ring ethylamine, to which it is attached COH group. So this histidine in presence of L-histidine decarboxylase enzyme converts to histamine. Now we know decarboxylation means removal of this COH group. So what happens whenever COH group is removed in presence of this enzyme, we get histamine. Now, as we have seen, this histamine degrades by two pathways. One is methylation, which is shown by this pathway, and second is oxidation, which is represented here by this pathway. So, histamine, first let us see methylation pathway. Histamine in presence of N-methyl transferase converts to N-methyl histamine. Now, as the enzyme name suggests, N-methyl transferase means methyl group would be transferred or attached to nitrogen. So this nitrogen of histamine, to it is attached methyl group. So it results in N-methyl histamine. This N-methyl histamine in presence of monoamine oxidase B. Now we know monoamine oxidase, what does it do? It would oxidize any group. So here this CH2, it gets oxidized to COH and we get N-methyl imidazole acetic acid. So this whole pathway is degradation of histamine by methylation. Second, let us see its degradation by oxidation. So what happens here? Diamine oxidase, what will it do? It will oxidize this CH2. So we can see here this CH2 is oxidized to COH. And what do we get? We get imidazole acetic acid. This imidazole acetic acid, again in presence of phosphoribosyl transferase, would transfer the ribose group. Where would it transfer the ribose group? To this to this nitrogen. So in presence of this hydrogen, here we can see ribose group. So this whole pathway is oxidation. So here we can see this is the synthesis of histamine, which is synthesized from histidine, and these two are the degradation pathway. First is methylation and second is degradation. Now moving ahead, let us see how this histamine is released inside the body upon any allergic reaction. So whenever any allergic reaction takes place, what will happen in any allergic reaction? It may be due to any dust particle, it may be due to any antigen. So what happens? This antigen, it binds to a specific antibody which is present in mast cell, that is IgE antibody. This Ig body is attached on the mast cell with the help of Fc epsilon receptor 1. Now we know that inside this mast cell what is present are histamine. So whenever this allergen binds to this Ig antibody, a sequence of events starts taking place inside this mast cell. First of all, tyrosine protein kinase gets activated. This activated tyrosine protein kinase, what does it do? It phosphorylates the IP3. So what happens? PIP2 is phosphorylated to IP3. PIP2 is phosphatidylinositol biphosphate and IP3 is inositol triphosphate. Now what does this IP3 do? It generates the intracellular releases of calcium. Now, as intracellularly calcium is released, it triggers the release of stored histamine from the granules. So, we can see that these granules are released and inside this granule is present positively charged histamine along with its negative counterpart as we had seen in the previous slide. This histamine is released from these granules with the cation exchange. So sodium and calcium catanically exchange histamine and results in release of this histamine. So this is the sequence of events which takes place upon which the histamine is released inside the body. Now let us see what are the stimuli which results in the release of histamine in the body. Like we have already seen, there can be any tissue damage, like it may be trauma, it may be stings and venoms, any proteolytic enzyme or phospholipase A. Second, there can be any antigen, like antigen antibody reaction which are involving IgE antibodies. Third, it may be due to any polymers such as dextran, polyvinyl pyrrolidine. Then fourth, it can be due to any basic drugs which we are taking such as tuberculin, then morphine, atropine, or finally it can be due to any surface active agents. So these are the substances which triggers the release of histamine inside our body. These are the five substances. These are also known as histamine liberators, which results in release of histamine from our body. So these are the references from which we have taken this. So we have seen in this slide about the synthesis, release and storage of histamine. Thank you.